so this is lab 2. It's only one exercise, but as you can see, it's a lot of information, right? It's examination of eukaryotic microbes. This is the only time we're ever going to, all semester long, work with or look at eukaryotics. The whole rest of the semester, anyone know what cell type we'll be looking at? Prokaryotes. Specifically, which type of prokaryote? Anyone know? Bacteria or archaea? Bacteria. Right, we're going to look lots of bacteria. Okay, so I'm not going to go over these terms, right? Well, but I just remember I said I'd always give you guys a glossary of terms. So they're here, they're in the PowerPoint. I might refer back to them if I need to. Okay, so there are two cell types that scientists talk about. They talk about prokaryotes versus eukaryotes. Pro meaning before, karyote a kernel. And as you can see, and already maybe have saw today, eukaryotic cells, this is actually from somebody's mouth, their cheek cells. So if you've taken A and P, these are what type of cells? Epithelial cells? Look like a fried egg? Nope, starts with the squamous, right? Squamous epithelial cells. So this big, large pink cell in that pink or reddish nucleus, yep, those are eukaryotic. So they have what is called a true kernel. It looked like a kernel to early scientists. That's where we got the name U for true, karyote kernel, right? We now refer to that as the nucleus. Bacteria, which are all the little purple specks that you see here, which are also in your mouth, right, as we've seen by our plate, are prokaryotic. They don't have a nucleus. And as you can see, they tend to be extremely small as compared to eukaryotes.
Some of those bacteria are good, some are
Bad, right, yeah. These probably not good. These are the ones that lead to tooth decay when you eat sugar. So if you're a sugar fiend like me, you just brush your teeth all the time. Make sense? Okay. So when we classify organisms, it all started with Carl, I always butcher his last name, Leonidas. Thank you. Linnaeus. So back in his day, really, you know, they were just starting to try and understand the world and categorize it. So really it was just two main groups, plants and animals. So then, when we started seeing other organisms that really didn't fit into that plant or animal category, we had to come up with more categories, right? More classifications. So what is the organization of the groupings that we put organisms into? Anyone know? Domains? Kingdoms? Anything else? Phylums? That's an unofficial group, right? So what we learned for life, right, for living organisms, plants, animals, bacteria, archaea, fungi, protozoa, we use the system now that starts with domains, and there's 3 domains, and then 5 or so kingdoms, and even some of those are changing. We're getting things like super and sub-kingdoms, and then as we narrow it down into phylums. So you can see this example here, right? The animal kingdom contains all the animals, right? And then we would go into a next grouping of a phylum. In this particular example, for this bear down here at the bottom, is Chordata. Anyone know what the defining characteristic of the Chordata phylum is? What do they all have? A spine, a spinal cord. Chordata is referring to the spinal cord. Mammalia, right, which we are too, right? We're mammals. What's some defining characteristics of mammals? Well, what are breastises actually? Mammary glands. So mammals have breasts, mammary glands. They're also covered in hair, right? They usually give live birth. Carnivores, what does that mean? They eat other animals, right? Meat eaters. This is the bear family. I can't even say that name, right? And then the genus, and then down to the single species. But when we say the species, we say both the genus and species name, right? We always capitalize the species name. We never capitalize. We always capitalize the genus, never the species, right? It's always lowercase. How else do we usually write the names of organisms? The proper name. How do you notice what's different about the type you see here? It's italicized. And if you were handwriting it, what would you do? The name. Proper name. You would underline it if you were handwriting it. If you were going to be accurate. And denoting that it was a proper name, both genus and species of an organism. Anyone know what our genus and species name is for humans? Homo sapiens. Capitalize the H, not the S. So, how are these groups determined? What did we just talk about? Well, how would we generally refer to, what do we consider having hair and mammary glands and a spinal cord? Traits, right? Characteristics. Is that the only thing that we use nowadays to group organisms? Anyone know anything molecular that we look at? DNA, and actually ribosomal RNA. That component of the ribosome that every cell has to have ribosomes, because ribosomes make what? They read messenger RNA to put amino acids together to form proteins. So it was through that we actually came up with the domain system, above kingdoms. And there are 3 domains. Which domain are we going to look at today? Yep, the ones with the nucleus, which the domain name is very similar to the cell type, which is eukarya. So when we look at characteristics, though, it can get kind of confusing if we were to just look at that one thing. Because all these guys have something in common, don't they? So this is a whale, a shark, and a fish. What do they all have in common? They live in the water. They don't all have gills. Whales don't have gills, right? They breathe air. They all have fins, right? I want my Venn diagram. So they live in the water, they have fins, and they can swim. But fish, definitely as compared to whales, are very different. Fish have scales, whales have hair, because whales are actually, what? Mammals. They give live birth, and they have to breathe oxygen via their blowhole. Where fish have gills, so do sharks. But sharks are even different from fish, right? Fish lay eggs, and of course they have scales. So some of us learn little sayings known as mnemonics to help us remember things. So a saying that another friend learned for remembering the classification scheme of kingdom, phylum, class, order, family, genus, and species is King Philip came over from Germany swimming. The one I remembered as a kid was King Kevin, please close our front gate slowly. Some people have family's gate, right? And domains come above that, right? So I've changed my sentence to, damn, Kevin, close our front gate slowly, right? D for domain, which comes before kingdoms. Again, pneumonic, not to be confused with other pneumonics like pneumonic plague, which is caused by a bacteria. So taxonomy is classifying organisms. The good news for you guys is this is constantly changing. So I want you to be aware that we classify organisms. I don't want you memorizing any of the classifications for any of these. So even though we have it listed to organize the lecture, you don't have to worry about any of these crazy names I'm going to say. The only name you need to know is the name of the organism we're looking at. Right? And just its genus name. Because not all of them do we have it down to the species. So phylogeny is a little bit different. That's studying the evolutionary process. Right? Where did these traits come from? Ontology, the course of development within a particular individual organism. And so sometimes ontology and phylogeny may be similar to each other, replicate each other, or they don't. Right? So sometimes how an organism develops gives us some insight into its phylogeny. So probably the best example of that is, anybody know a feature that we have when we're developing in our mom that we actually do not continue to develop and we're not born with? A tail. A tail, right? But that gives us some linkage to the other mammals that we're familiar with, right? Like cats and dogs and things like that. That actually, and monkeys, that actually use their tail. Right? So you could almost think of what we're trying to construct is, for all organisms, their family tree. Right? Where did they come from? We do this for people all the time. Right? We can look at these two guys down here that have this common ancestor. One of my uncles did our family tree for the Mabe side. Goes all the way back to England. He was a history professor here at Delgado. He's retired now. He'd like to tell me all the time that we were here during the Revolutionary War. That family. I was like, okay. It's an old family from England. So again, when we start trying to tease out like where they came from, you know, there may have been a common ancestor somewhere here in the beginning, and we diverged off onto two paths, bacteria or archaea, these single-celled prokaryotic organisms. But in forming things like Protista and Plantae, there may have been some merging. So an early archaea merged with the bacteria to form Protista. There's pretty strong evidence that the organelles in a lot of the eukaryotes actually were once free-living prokaryotes, whether it be bacteria or archaea, that were engulfed by the early eukaryote and began a symbiotic relationship. And then over time, that progressed so much that they couldn't live without each other. And we now know these as organelles. We have pretty good supportive evidence that two major organelles that are found in eukaryotes were once free-living prokaryotes. Anyone know which two organelles? So, nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, chloroplast. Mitochondria, he's right. The other one is chloroplast. Because if you look at these, they have their own DNA, they have their own ribosomes, and guess what? Their ribosomes look like prokaryote ribosomes. So there's pretty strong scientific evidence that these were once free-living organisms that were engulfed by others. You actually get your mitochondria from your mom, not your dad. Not really, because you can get a genetic disease because... All right. So this is our tree. This is based on the work by Carl Woese and George Fox. Poor George, he's the second name. He usually gets dropped. That came up with this 3-domain system by studying and analyzing the ribosomal RNA sequences of organisms. So again, we're going to focus in on one domain today. That's eukarya or Eurycata. So where are humans? What are we? Which domain? Eukarya. Which one of these would we fall under? Animals. Yeah. We put ourselves on this pedestal and we forget sometimes, right? We are animals. So then, when we go into these super groups, as I said, right? So we've gotten, even out of the kingdom classification and all of that. There are either 5 or 6 pretty well recognized now supergroups. So when we look at our tree, right, this is the 6. Our book goes over just 4, so these 2 are usually combined into Uniconata. Uniconata. These names will kill me. So we're humans now. We're somewhere on this tree, right?
Do we have to memorize that?
No. You should know we're animals, though, right? We're Metrozoa. And there's a really cool website that Berkeley put together, if you want to check that out. So for us, is this stuff you need to know? Yeah, this is on your homework sheet, right? If you don't have these filled in, you might want to fill them in. So for prokaryotes, there's 2 domains, right? Which of the 2 are prokaryotic? One starts with an A, one starts with a B. Bacteria and archaea. What domain are we looking at today? Eukarya, which is comprised of all eukaryotic cell type. Some kingdoms classifications that we're still kind of using, you're probably familiar with 2 of them, plants and animals. That's easy, right? But this is microbiology class, so we're going to focus on protists and fungi. Other groupings that you'll hear, but not true classifications. Protists are usually sometimes divided between protozoa and algae. In fungi, we usually say whether it's a yeast or a mold. So although these are not classifications, these are unofficial groupings that are used by most microbiologists. So you'll hear me use them a lot. So we're going to start at Excavata. We're going to look at a representative in this group, Parabasilidia. Anyone know what that organism is? You will after today. We're going to look at one in this group. Anyone know who this cute guy is? No, it's Giardia, and I got a poster in the back. And remember, this is the one we don't have good slides for, unfortunately, because he's cute. He looks like he has eyes, huh? It's paired nuclei. And then up in this branch, we're going to look at Euglena. And this you could find in pond water. It's not parasitic. And then we're going to also look at Trypanosoma. So this group, what they have in common, and again, you don't have to memorize this, but it's good to, get a general idea why these guys group together. They're all unicellular. They're single-celled organisms. They all have a feeding groove. They have 1 or more flagella, and we've got some really good slides where you guys will actually be able to see the flagella for the Trichomonas. Some of them have a life cycle where they're trophocyte and a cyst. Does anybody know what the cyst is? What, Chris? Giardia actually has a resting state. It's a resting state. I answered my own question. Okay. Where the trophocyte is the feeding phase. So some of them can protect themselves in the cyst form. They're not feeding, they're resting. And so you could still ingest that and get infected. And that's definitely true for Giardia. Again, I don't bother having you guys look at the cysts. they can be really hard to see under the microscope. So we're going to stick to, if they have two different phases, we're going to stick with the feeding trophozoite phase. So the first one we're going to look at is Trichomonas vaginitis, and this is one of my new slides. And this is a special expensive slide we just bought that actually has the flagella stained so that you can see it. So you should be able to very easily see the nucleus and the flagella. Of course, you're not going to see the undulating membrane undulating because they're prepared slides. They're dead. Only know to have the trophozoite, no cyst phase. These are fermenters incapable of oxidative phosphorylation. So they don't have an electron transport chain like we do. They can only ferment things. They have hydrosomes, which produce hydrogen gas under anaerobic conditions. So these guys are really smelly, and they cause a sexually transmitted disease. This you do need to know.
If it's fermented, it would be like a yeast infection?
No, but it can cause an infection of what area of the body? It's a vagina, and it's sexually transmitted, right? So this is trichomoniasis, right? Sometimes referred to as trich for short, not to be confused with trichinella, right, which is a worm, or trichiureus, which is another worm, right? Often, but not always, causes an inflammation of the vulva or even the vagina for women. Yes and no. Yes and no. And sometimes not knowing that they have the infection and transmitting it. And men can be carriers, and they don't show any signs or symptoms. So then they don't know that they're transmitting it. Yes. No, it's not. The yogurt's not going to help you either. It's a protozoal infection. Okay, so the next one is Giardia. You do need to know about him, right? Unfortunately, the picture even in your book is really bad. You can't even see his flagella, right? Can you see them? No, you can't. But he has 4 pairs of flagella. He's found in the trophozoite as well as the cyst phase. He has a non-functioning mitochondria, so he relies just on glycolysis. Paired nuclei, which is why he looks like he has eyeballs, but he doesn't. He has 4 pairs of flagella. 2 medial bodies, so sometimes also he looks like he has a smiley face. As I said, it does form cysts, and it's resistant to water treatment. It can commonly referred to as beaver fever when you drink stagnant water from ponds. And nowadays, most people refer to it as Giardia instantalis. It can cause an infection of the vagina, the labia, and stuff like that. That's where it had gotten that name. But turns out there's just one species. So at this point, hopefully the transition will happen where everybody's on the same page. Yeah, so you could ingest this, right, and it causes a really serious form of diarrhea. So the other one we're going to look at that's a new slide is Euglena. So these guys are really cool. They're green naturally because of their chloroplast. They have 1 or 2 flagella. On these slides, I'm not sure if we'll see that. They have an eye spot or stigmata, which helps them find the light as they swim through the water. These guys would live in aquatic environments like our pond water. They can photosynthesize, right? They're autotrophic. They can use light energy and carbon dioxide to make their own food when light is available. When it's not, they'll eat other organisms. They'll be heterotrophic. They'll engulf and eat other organisms. So this is one of those guys that, it's like, well, is it a plant or is it an animal? Well, it's kind of its own classification, right? It does both. So what structures give these organisms their photosynthetic ability? What allows them to process the light? The chloroplast. So they have chloroplast, and that's why they're green. Because most of the green stuff you see inside of them are their chloroplasts. So like all these green spots, it's chloroplast. And that little red spot is that eye spot. Helps them find the light. So the last one is the trypanosoma. And so in your book, these guys have one enormous mitochondria. They have a kentoplast, and I'm going to have another picture that shows up with arrows, and that's what the arrows are pointing to. What disease do they say in your book this parasite causes? African sleeping sickness, right? But that's Trypanosoma brucei. And there's a subspecies, Gambesia or Rhodesium. And of course, this is a problem in Africa. It's transmitted by the tsetse fly, this protozoa, right? It's injected into your blood. This is the protozoa. These are red blood cells. So for these samples, you'll see red blood cells and you'll see the parasites. You're looking for the parasites, of course. The slides that I bought, because it's more related to us here in Louisiana, are the Trypanosoma cruzi, which they don't talk about in your book. These cause Chagas disease, and we do have it here in Louisiana, and it's transmitted by the kissing bug, if you've heard of it. And there's been some dogs in the swamps and stuff that have gotten this disease, right? And they're still trying to figure out what they can do to treat it. Yeah. The cruzi one is the one we're going to look at. And apparently, under the microscope, these guys look relatively similar. So there's the nucleus.
We have that in Louisiana?
Yeah, this one, cruzi, not brucei. This is African sleeping sickness. This is Chagas. The kissing bug transmits it. I forget the actual name of that bug. I'll have to look it up for you guys. No, it's not love bugs. It's not love bugs. And in fact, guys, if you go to, like where I steal my pictures, I always put in my notes section. So this website right here, tells you all about it.
Both of those cause sleeping sickness?
Mm-mm. So this one is actually a case study about and how to differentiate the two. But I thought, I guess it was a different page I saw. I should add a picture of the kissing bug and the actual scientific name of them. But this is one of the new ones you guys don't have to know just yet. You just need to know Trypanosoma brucei causes African sleeping sickness. Trypanosoma cruzi causes Chagas disease.
Which one do we not need to know?
You don't need to identify this one under the microscope, but you need to know the two different trypanosoma and what diseases they cause.
And the cruzi causes what?
Chagas disease.
What is that? Disease.
Look it up. Okay, so apparently the difference is this part right here that I have the pointer pointing to. For ours, it's going to be relatively large. Don't confuse that with the nucleus, although that it does contain DNA. That's the kentoplast, and this is the nucleus. But as I said again, I'm not going to ask you to identify these under the microscope. Just be aware of them and what diseases they cause. All right, so this is our summary slide. So this is where I'm going to pause. This is the new slide that you guys will have access to when I post the new version of the PowerPoint. Is work together as a table, right? Remember this one, Giardia, we don't have slides. But I'm gonna pull out my new slides for all three of these guys. I got another set of trays I'm gonna hand out. And we can go ahead and look at these. You know, one person can focus it and then everybody can look at it. So if there's 3 of you at the lab bench or even 4, you know, you can look at another one. I'm thinking low power for Trichomonas. Probably low power for Euglena. Trypanosoma, this one I would say low power, but depending we may want to go to oil. We'll see. We'll see how they look. I haven't looked at these in a while, so I'm not sure. Found it. All right, so let's go to the next group. So this group, we've got, this is the last one on the new set of slides, I think. This is Chlamydomonas. This guy is really cool. You see he's got flagella and a chloroplast. I can't remember if they stained him and made him not pretty green. We'll see. These are Volvox. These are supposed to be green again, but they've stained them, so we have purple ones and pink ones. Look like little balls with little balls inside of them. The individual cells, we don't have the ability, right? These specimens are too thick. Even if we went to oil, you wouldn't be able to focus in on the individual cells that you see in this picture. And then everybody's favorite, because it's easiest to remember its name, good old Spirogyra has chloroplasts on the inside that are spiral. Thankfully, they did not stain these slides, and you will see the green. Because guess what all of these guys have? Chloroplasts, right? They all can photosynthesize. They're all autotrophic. They can use carbon dioxide from the atmosphere to make their components. They all have cellulose in their cell walls. This sounds very similar to what other type of organisms we know about that photosynthesize? Plants, right? So these are your microscopic plants. So the first group, these guys actually live in water, hence the flagella, right? They can be unicellular like the Chlamydomonas, or they can be filamentous or colonial, where they live in a group like the Volvox. So we're going to look at Chlamydomonas and Volvox. And then the next group, which is the very closest relatives to plants, is our Spirogyra. So good old Chlamydomonas, one big, huge chloroplast. These guys are haploid. Anyone know what that means? They're not diploid. Well, what does diploid mean? Two sets of chromosomes, right? One you get from mom and one we get from dad, right? These guys, no mom and dad, and just one copy. They're unicellular. It's a single cell. It has a chloroplast, right? It has two flagella. They have a stigmata as well, so an eye spot to help them find the light, to be able to move towards the light. As I said on the videos page, check this out. This guy's hysterical. Sings a song about Chlamydomonas. Again, don't confuse Chlamydomonas with things like chlamydia, which is a gram-negative bacteria, or things like conjunctivitis, or Neisseria gonorrhea for that matter, right, which is another sexually transmitted disease, sometimes referred to as the clap, right? Why? I do not know that one. Why? Oh, nice. Okay. I really didn't need to know that. Okay. Now I know. Do you know why they say people were dirt poor?
Does they eat dirt?
Nope. Their floor was made out of dirt. There's a meaning behind these sayings that we say. They came from somewhere. No. Well, I don't know. Depends. Okay, so we're going to be looking at Volvox, right? This is on that original set of slides I gave you. You're going to be looking for these big balls, these colonies of cells, and then they form smaller colonies inside the larger mother colony. These are called daughter colonies, right? So these are colonial, as I said. They can reproduce sexually. They'll produce sperm bundles and female colonies where all of the cells are eggs. Then the spermy guys swim over and they fertilize, right? But usually they reproduce asexually. So what type of reproduction are the daughter colonies? It's asexual, right? This is a really cool website on Volvox where I got most of these pictures. So as I said, we're not going to see the little guys, we're going to see the big colonies. And they are colored pink and purple. Spirogyra, we've got two slides of this one, two prepared slides. One of them you can see the nucleus clearly. You can see the spiral chloroplasts. These guys are filamentous, right? So they form long filaments, but they're made up of individual cells. You can see the cell wall between the two cells there. They also are haploid, but they can conjugate. They can join up with each other, right, and become diploid. This sexual reproduction is referred to as conjugation. And we have one very expensive slide that shows this process. And the final step where the spore that's diploid that is formed that will bust out of here and form a whole nother set of filaments that'll be genetically different, right, because two of them combined. So one of your slides has the word conjugation, I think, on it. And you'll see it shows this process of two of them coming together and exchanging that information. That spore is called a zygospore. And so you need to be able to identify the chloroplasts, right, which are spiral, the nucleus, and the cell wall that divides each cell. So this is the summary of the ones, right? So Spirogyra, you definitely need to know. This is an excellent drawing I found on the internet, and again showing the conjugation. The Volvox, again these guys are stained. We're going to also look at the Chlamydomonas. I'm not sure what magnification. This is the new one you don't have to worry about identifying, but go ahead and take a look at it. So the next group are some of my favorite ones, especially if we had some live specimens. Alright, so these are some pictures of the ones we're going to look at. I'm going to skip over this. This is just FYI. So remember I talked about that endosymbiosis we believe that happened, that a prokaryote like a cyanobacteria, which is a photosynthetic bacteria, was engulfed by an algal cell, and basically cyanobacteria became the chloroplast. They believe that algal cells were then engulfed by these other cells. So their chloroplast, or sometimes referred to as the plastid, is the result of two levels of endosymbiosis that happened. Still with me? So that's the first event, right? And then it got engulfed again. And it's the modern day chloroplast you see in some of these cells, or plastid. So these guys can either be heterotrophic, eat other organisms, or they can be autotrophic. And actually I think most of the ones that are on here, except for the diatoms, are heterotrophic. So paramecium eat other organisms. You've probably looked at paramecium before, right, in biology class. Centaurs is one of the new ones we have on our list. Thank you. And so not responsible for yet. These guys are super cool. I love to watch the live specimens swim around because these guys have cilia. And again, you're not going to really see that well under the microscope, but there's really cool YouTube videos I have linked. Bellotinium coli, again, this one is actually parasitic. It'll cause a really bad infection for you. And then Plasmodium falciparum, this one's not motile and has several different phases to its life cycle. The good news for this class is you just need to be able to identify the parasite as opposed to the red blood cells that'll be on the slide. Anyone know what disease this causes? Plasmodium falciparum. This you do need to know. It's transmitted by the bite of a mosquito. Malaria. And then diatoms, although this group usually contains ones with the flagella, these guys do not have a flagella. So paramecium, as I said, most of us have seen this. Remind me next week, y'all. The biology lab gets in live cultures. If you guys want to see the live ones moving around, you got to remind me. I'll go steal them from biology. They should have a macro and micro nucleus. Usually we just see the macro nucleus in our slides, not the smaller one. They have cilia. Again, we don't have the fancy-dancy slides like this picture where the cilia are stained. It's going to be really hard for you to see them. You should know that they have cilia. Again, depending on the slide, you may or may not see the oral groove. They have contractile vacuoles. They're really fun and awesome to watch the live organisms continually squirt the water out that comes rushing in. So I have a couple of links to some cool videos for you guys to see those characteristics. Centaurs, again, these are ciliates covered in cilia. They swim around, but they can also attach to their surface. So most of the time they're going to be this really pretty trumpet shape, but they can kind of scrunch up too. So they're going to look all different on your slides because they're an, suspended animation. They have beaded macronucleus, right? So these dots all throughout, that's their nucleus, macronucleuses, more than one. The centriome is that little white spot you see here in the dark coil. It's where they take food in. It's like their oral groove. They're naturally green. I think they stain them for these slides too. So these guys are naturally red, and the slides aren't stained, so they are this nice, pretty pinkish red color. Belatinium coli, you can really identify them very easily by the shape of their nucleus, which is very distinct for them, this kidney bean shape. We're just going to look at the trophocyte, the feeding phase. Again, they have cilia, really hard to see. Kidney bean shaped nucleus. What are some of the symptoms if you had an infection with this, if you ingested this? Yeah, you're going to have bloody mucoid stool, your feces. You're going to have diarrhea, maybe as often as every 20 minutes. Over long term, you'll have alternating diarrhea and constipation if you have a chronic infection. Mostly this is a problem in developing countries, right, where the water gets contaminated by pigs, swine, and human feces. Plasmodium falciparum, this is 1 out of 4 species of Plasmodium that causes malaria. So again, we have slides that have red blood cells, and then the parasite will either be inside the cells, and I really want you guys to try and find ones inside, or even outside. And they're going to have different forms to them, right, the parasite. But the only thing on those slides should be the red blood cells and the parasites. So differentiate parasite from red blood cell and try to find some with them inside. And we already said causes malaria. So this is the big bad one as it relates to malaria, this particular species, falciparum. So transmitted by the mosquito, has a life cycle. The good news is you don't have to know all of this for this class, but it's here. For those of you guys continuing on in medical fields, it's good to know this information. Where is this prevalent? Where would you travel to and get this infection? Africa, right? Here's some other biters we want to worry about, right? And definitely here, West Nile, this particular mosquito. Dengue and yellow fever, which yellow fever we've had in Louisiana. It's not here right now. It could come back. Dengue, I always say that one wrong. A student of mine had that. I have a poster over there.
That's really dangerous.
Yes, not good news. So mostly Africa, but South America, right? And then of course we have good old Zika we're scared about now too. Transmitted by mosquitoes. That's a virus though. And then, you know, parts of Southern Asia. So it's an interesting phenomenon as it relates to sickle cell anemia and malaria, because sickle cell, partial sickle cell is protective against malaria. So if you're not aware of that, that's an interesting read. I talk about it in my genetics class. So diatoms are the ones we're going to look at next. And so these come in 2 forms. These are sometimes referred to as nature's stained glass. These are photosynthetic, right? Aquatic brown algae evolved from red algae. Single-celled or colonial. Again, they can use carbon dioxide from the atmosphere. They lack a flagella, unlike most organisms in this grouping. They have 2 halves that overlap like petri dishes. Can't really see that with the light microscope, those high-powered microscopes. You can, if you search on Google for images of diatoms, oh my gosh, there's so many gorgeous images of high-powered microscopes, and they're just beautiful. So we need to know the 2 different forms. The centric, right, which is the radial symmetry, spherical ones, as opposed to the pinnate, which are the bilateral or elongated ones. So I want you to be able to try and find both. Now these guys are sometimes called nature's stained glass because they contain what substance in their cell walls? What makes up glass? Anyone know? Silica. Or silicon dioxide. Right. So go ahead and get your mixed diatom slide and see if you can find both pinnate and centric. Right. So circular versus elongated. So the last group, Unicaudata. So, no, is this the last group? I hope it is, yeah. Almost. So the amoebas, we just have the one amoeba. We don't have the entamoeba histolytica. So just these guys, again, they're stained all one color, either purple or pink. This is actually a live one. And again, remind me next week. I'll go get some and we can watch them move, hopefully. So freshwater, free living, you need to identify their nucleus, their pseudopods. They don't have a cell wall, obviously. They wouldn't be able to morph like they do. So sometimes referred to as false feet. And so again, here's a YouTube of it actually moving. It's actually a video I took in lab. And then others of them feeding. This one we don't have, right? The slides are poor. This one, it's again going to have an irregular shape, but how you identify it is this very unique nucleus it has, this bullseye shape, right, where you have a dot with a ring around it. So this one, again, there would be a stool sample. That's why, again, for beginning scientists like yourselves, or microbiologists, it's really hard to distinguish the poop from the organism. You have to really look for that nucleus. And they weren't well-stained slides, so we've given up taking them out. So then we have the fungi. And so we have yeast. That's what you see here. And some of you guys looked at these already because those were a good one to do oil. And then the unofficial grouping among fungi, we say yeast, and then what else? The multicellular ones. Molds. These are my favorite. We save the best for last. Right? So these guys are non-motile. They contain cell walls that contain chitin. Yeasts are unicellular. Right? They bud. So we're going to be looking for their little baby buds. That's asexual reproduction. But some of the times their buds will stretch out and it'll almost look like hyphae, which is what you see in molds. So it's not true hyphae, so it's called pseudo-hyphae. And you definitely see this with our older slide of Conidia albicans. Molds, on the other hand, are multicellular, right? We can see them growing on our bread and things like that. That big mass is called a mycelium. Under the microscope, the individual fibers we'll see, right? So this is the big mass. The individual fibers you see under this microscope, that's the hyphae, like the filamentous-like cells. So these guys are classified by their sexual spore production and the presence of cross walls in their hyphae for molds. So we have 3 species we'll look at of molds and 2 yeasts. So Saccharomyces, anyone know what we use this one to make? Beer, wine, bread. So, does it cause disease in humans? No. This is the one we like. So these are teeny tiny, oil immersion. Conidia albicans, one of our older slides, you'll see these long threads, that's pseudo-hyphae. But you'll also see them budding. It depends on temperature for these guys. And this one does cause an infection, commonly known as a yeast infection, which most women would get in the vagina or vulva region, but people can also get it in their skin. And this is when the yeasts that are naturally present, we all have these, overgrow. And they can overgrow for several different reasons. If you're lucky enough to have never experienced a yeast infection, the rest of us are jealous. So just again, some more examples of what they would look like under the microscope and the different parts. So my favorite, the molds, right? So under the microscope, they're quite gorgeous, right? When we look at their spore structures, that's how you differentiate the different species. So again, this is what it looks like on an agar plate. This is a slice into the agar even, and looking at it under the microscope. So these very thin threads that you would be able to see are up above the agar, and down here in the purple region is growing into the agar itself, right? So you can't just cut mold off of bread or cheese, right? It grows into it, releases its enzymes to digest the food and then absorb it. So we're going to look at Aspergillus. We have brand new $15 slides that are really expensive that actually take these balls and cross-section them so they look like these beautiful flowers. So you can see the middle part that holds the spores. Each one of these little dots is the spores, right? That would be released into the air, land on another food source, and grow another mass of hyphae, a huge mycelium, and then they'd start growing the aerial hyphae, right, which are these sporangiums with the spores on them. So Aspergillus, this one is actually used commercially to produce citric acid, as well as it's used in soy paste and soy sauce to ferment the soybeans. And this is an interesting backstory about Aspergillus. So this is my favorite one. It's so pretty. Looks like little flowers. Penicillium is my second favorite one. Right? Its structures look like little paintbrushes, and that's actually where it gets its name. So those spores are called conidia. They're asexual. And then the mass, right, of fibers you can see down here at the bottom are the hyphae. And it produces penicillium, of course, penicillin, under stressful conditions. Under other conditions, it actually makes different cheeses and helps in the preservation of sausage and ham. The last one is Rhizopus. We have two slides for this one too. One that shows asexual. So here's the spores in the little cap region here. And the other one was sexual, where the two come together and form the zygospore. And this you would find on your bread. This last one is what you would find on your bread. So here's some detailed drawings and labelings of what we want to look for, right? The Aspergillus, we have the two slides. One actually shows the pretty cross-section, so pretty little flower-looking things, right? The Penicillium, again, these long, almost brush-like looking. And for Rhizopus, we have the asexual with these little caps, and the sexual where it comes together. It almost looks like the spiky balls that fall from the sugar gum trees. You guys know what I'm talking about? Those little spiky. It looks like this. See how it's spiky? Right. So make sure you guys look at those. This is my last slide. Other than I have a life cycle one, again, just for information. So I'm going to go ahead and